Oh man. Ew, I can't do this. Good morning, everybody. I have to go over here, otherwise I'm completely blinded by the light back here. Welcome back to the human health session, the continuation. I hope you had a wonderful evening in this beautiful reception yesterday. And today we will continue with examples and tools. And the first two speakers will present on the BPA studies. And today we do it a little bit different. So we have question and answers directly after each presentation. So without further ado, I would like to introduce Dr. Barry Delclos, who is a research pharmacologist in the Division of Biochemical Toxicology at the US FDA's National Center for Toxicological Research. And he will present data on the Clarity BPA project, the core NCTR-NTP study on BPA. Please.
Thank you very much for the introduction, and thank you for the invitation to speak. So when I was given guidance on this, preparing this talk, I was told this wasn't to be a session on BPA toxicity, but rather on using this Clarity BPA project as a model or for a cooperation between government scientists, including regulatory scientists, and academic scientists. So actually, I'm not going to be presenting data from the study, but I'm going to be talking about how the study came to be and how we interacted in terms of developing the core study. And I'll explain what the core study is in a few minutes. So I'm going to first talk a little bit about the background on BPA research conducted at NCTR, which you heard much about yesterday, actually, in terms of the pharmacokinetics. And then the conception and implementation of this Clarity BPA program involving how the academic projects were selected, the planning of the core study design, interaction with the academics, and then compromises that had to be made along the way. And then I'm going to talk about the study, the conduct of the study, of the main study, and the reporting process for the main study. And one of the things, there were good things about the project, there were bad things about the project, there are things that we've learned to go forward, and that'll be the subject of the next talk. So this is a slide for anybody who's been asleep for the last 20 years. BPA is used to make polycarbonate plastics and epoxy resins that are used in can linings. There's widespread low-level exposure, and it's regulated by FDA as an indirect food additive that migrates in small amounts from the food packaging. The use of BPA is decreasing with time. So there's been considerable debate over the risk posed by low-level exposures. And the guideline studies, the large guideline studies in mice and rats showed no effects at low doses. But there are many studies out there which indicate that report that BPA induces a variety of effects in a variety of model systems at relatively low exposures. And these effects often reported to occur, so that the high dose effects do not predict those that are seen at low doses. Much of the work, certainly the early work, has been done on the assumption that bisphenol A is a weak estrogen, and it's acting as a weak estrogen agonist. But now there are multiple other mechanisms of action that have been invoked to explain the reported actions of BPA. So this gets us started as to when NCTR started working on the project in 2008. Currently the status is that most regulatory agencies around the world indicate that BPA is safe at current exposure levels, and that is a subject, has been a subject of controversy now, as it was in 2008. And then in 2008, there were two reports, one from the NTP on the risks of human reproduction, and also a draft safety assessment produced by the Center for Food Safety and Applied Nutrition of the FDA, who have responsibility for regulating foods. The FDA Science Board, which is an outside body that advises the FDA, reviewed that draft assessment and made numerous recommendations that should be pursued. And some of the key recommendations were that a rodent study should be considered, and I'm not going to read all this, but to meet the criteria established by FDA or reasonable criteria, to address the endocrine mechanism-based concerns, and to use endpoints and models validated for the study of endocrine-mediated development. And then also, importantly, to develop BK models to allow for extrapolation across species. And the research program was constructed as followed in all the top material, the pharmacokinetic studies, physiologically based pharmacokinetic studies. A tremendous amount of data was generated, and that was described by Dan Dorgy yesterday. And what I'm going to talk about a bit were the toxicology studies, and particularly there were several different toxicology studies conducted at NCTR. I'm going to talk about the guideline compliant GLP studies that I was involved in. Okay. So we started by a 90-day subchronic toxicology study, and the model we chose was a Sprague-Dawley rat from the NCTR colony. It's been maintained there since 1972. It was used by researchers for a variety of purposes throughout the center. We had used it previously in studies that we conducted in conjunction with the NTP, multi-generation and chronic dietary studies with genistein, nonylphenol, and phenylestradiol. Actually, it was genistein and phenylestradiol were the two compounds which included a chronic component, a 2-year study. And also, importantly, they were used in Dan Dorgy's studies in the comprehensive evaluations of BPA pharmacokinetics across life stages, and that informed some of the decisions that we made going forward. So we selected housing conditions different from those that are generally used at NCTR, and diet to minimize the exposure to BPA, and to other xenoestrogens. We monitored for genistein, coumestrol, zearalenone. We also monitored BPA levels in extracts and cages, bedding, water bottles, and feed. And we found consistently that the rodent feeds were giving us very low levels of BPA above the analytical background. And we established a cutoff. That was the only cutoff in the diet that we had established, that it had to be less than 5 parts per billion in the diet. And in fact, in all the studies that we've conducted, I think we only had to reject 1 lot on that basis. We used oral dosing by gavage 7 days a week. The scheme for the 90-day study is given below. We started at gestation day 6 after fetal implantation, and then continued dosing the dam daily, and then stopped at the time of the initiation of parturition, and directly dosed the pups from postnatal day 1 through the termination of the study. And the reason that we dosed the pups directly was information that we had from the pharmacokinetic studies that indicated that the milk transfer was very low, so attenuation of dose approximately 500-fold from what was given to the dam going through the milk and to the pup. So we had used a naive control, not handled, not dosed by gavage, a vehicle control. We used what we called, for the study's purposes, low BPA doses, where we went in half-log doses from 2.5 to 2,700 micrograms per kilogram body weight per day. And then high BPA doses that we would expect from the guideline studies to have produced some adverse effects, 100,000, 300,000. And we included reference estrogen controls. We collected standard endpoints and, importantly, the histopathology. Dan's lab conducted for us internal dosimetry across life stages, so that blood levels were measured at the Cmax and PND4, then PND21, the time of weaning, and then in adults. And there was also some gene expression and methylation studies conducted. I've just got a one-slide summary of those findings. We found no differences between the naive and vehicle controls and the endpoints that we had measured. We found that E2 and dose-dependent adverse effects in females were much more sensitive to the males. There was a high BPA-induced clear adverse effects that was overlap with the reference estrogen controls, and the females were more sensitive than the males. We did see some sporadic, statistically significant effects in the low BPA dose of sort of questionable biological significance, and those studies were reported in the papers that are indicated below. So, okay. So during the time that we were conducting this study, actually it was NIHS, the Division of Extramural Research and Testing, Research and Training, and specifically Jerry Heindel, came to the NTP and the FDA and suggested that we establish a collaboration with academic laboratories to bridge investigative and applied regulatory research. And this was proposed to be a consortium of NIH-funded investigators, along with federal scientists and regulators, people from NCTR, the regulatory scientists from the Center for Food Safety and Applied Nutrition, as well as the NTP and NIHS. There are two key components, the core chronic study and then the academic grantee studies. I'm going to talk about the core study development. So in 2010, there was a grant funding opportunity announced by NIHS, indicated that the purpose was to develop a consortium of researchers to work with us, with NCTR and NTP, in the final design of a chronic gavage study in the NCTR Sprague-Dawley rat, following development and direct dosing. So following the protocol that was used in the 90-day study. Proposals were solicited for hypothesis-driven mechanistic studies, and it could be added to the chronic study design. Proposals were reviewed and selected by an NIHS peer review committee in 2011. So we, as the persons conducting the study or the regulators, did not have input in the selection of these studies. We, NCTR, did have input as far as reviewing the proposals that were selected for feasibility. Could we conduct the work that was proposed? And we had to sit down with the staff, the large staff that was involved in animal care, pathology, and so forth, to discuss with them. And then we went to NIHS, NTP, and discussed the feasibility. There were no, I imagine that the proposals were reviewed and graded, given grades. We were not given, there was no information on prioritization. We tried to do as much as we could, get as much as we could into the study. And we did not reject any of the studies that were selected, although some modifications were necessary to many of the proposals. So the goals again were to evaluate chronic exposure over a broad dose range using both the traditional endpoints in our study and non-traditional endpoints, and determine if those non-traditional endpoints revealed toxicities wasn't indicated in the guideline study endpoints, and also to determine if these non-traditional endpoints could provide any mechanistic support for observations that were made in the guideline study. And there was an important component as a dose response assessment, again covering a wide dose range of BPA with reported effects in the scientific literature. So we followed the usual procedure. We have a long-standing agreement with the NIHS, NTP, interagency agreement in which studies are conducted, and we followed the usual format for that, so that we proposed a protocol and then presented it to the committee, the Toxicology Study Selection and Review Committee, which consists of NIHS, the FDA regulatory centers, and NCTR representatives in November of 2011. And we proposed to use the same animal model and dosing regimen, again, as used in the NCTR 90-day study. We had originally proposed to go up in tenfold dose steps from 2.5 to 250,000 micrograms per kilogram BPA. So we were basing this on a traditional dose selection paradigm using the 90-day study results to inform what we would do for the 2-year study. And we only saw clear adverse effects above 100,000 micrograms per kilogram body weight per day. We did propose to include a set of animals dosed continuously, and because developmental exposure is important, also what we call a stop dose group that was not only dosed until postnatal day 21, and then continued untreated after that. And we received tentative approval of the TSSRC pending input of the academic side. We had a meeting at NCTR in 2012 in March, and it was universally agreed that the high dose should be dropped because it was out of the range of real regulatory concern, and actually FDA agreed with that. One investigator did request that we include that high dose based on expectation of finding toxicity with BPA at that dose based on our 90-day study. Also, the academic investigators felt that we should include ethynyl estradiol as a control to demonstrate the sensitivity of the system under these study conditions. And the dose levels were informed by the 90-day study. The high dose that we had used was decided to be too high. We went to the 0.5, and then there was a tenfold dose lower that was used. There were a lot of practical considerations in the conduct of this study, and one of them was that we had to drop the number of animals used in the EE2 groups because we were simply, there were space issues as well as personnel issues. Okay. As I indicated earlier, there were no study was totally rejected, but some proposals did need modification. In most cases, the grantee animal sets included 10 animals per sex, per study arm, per time point. And in some cases, there were more that were requested. There were several specific requests that we couldn't meet. One investigator was insistent that for her studies to work, we had to have a subset of animals go on high-fat diets and had to do body composition by MRI, and that just could not be accommodated. And similarly, euthanasia methods could not be tailored for specific endpoints. At any rate, at the end of the meeting, a final protocol was agreed on by all the consortium members. I left out that second bullet there on the modifications because some things developed after the study started that there were a limitation of the number of animals that were produced where we had to make changes on the fly. And one of them was that we had to cut back on some dose groups of for the investigator doing the prostate studies, we could not have the subset of animals implanted with testosterone and estradiol implants. A second thing that had to change on the fly because of a lack of ability to supply both the core study and the grantee studies was we had to cut back on the number of animals that we had scheduled for the 1-year sacrifice in our study. We had originally scheduled for 26, but in order to meet the requirements of the academic investigators, we amended the protocol on the fly and provided the animals to them. Okay. So this is just a schematic of the way the study was done. The core study, as I said, was a modified guideline compliance study. We had a 1-year necropsy, which included a variety of standard endpoints, and a 2-year necropsy, which included only histopathology. Siblings of the animals used in that study were given to the grantees for various molecular, structural, and functional endpoints reported in the literature to be associated with BPA. And the animals were removed from postnatal days 1 up to 12 months, depending on the study. And all these endpoints are endpoints that were not typically included in guideline studies. Just briefly, the dosing regimen, we had a continuous dosing arm, same style used, same protocol used in the 90-day study, but we went out to sacrifices at either 1 year or 2 years. And we had this stop dose arm where dosing was stopped at weaning, and then animals were continued without treatment. I've added again this line about the direct dosing of the pups to remind myself that while we based this on the BPA, the same type of pharmacokinetics occurs with phenylestradiol in the rat. So the adult rat gets a very low exposure at a given dose relative to a pup that's directly dosed. So the adult, there's very low oral bioavailability of E2 in the adult rat. So the same high dosing, differential dosing, high doses administered directly to the pup at weaning versus the adult that we see in BPA is true for phenylestradiol. Okay. So I'm not going to go, a lot of this is covered in previous slides and is repeated, so I'm not going to do that. There were study elements that were common to both the core and grantee studies, including the dose levels. I mentioned the 5 dose levels that we ended up using in the 2 EE2 groups. And the point that I wanted to make from this slide was it was up to the grantee as to whether they wanted to take, just as we had, restrictions based on logistics, so did they in terms of the number of samples they could handle. And some grantees chose not to use both dosing arms or not to use all subsets of doses, and that was their choice. Okay, so we did do, for all the studies, identity and purity of the test articles. We certified the dose formulations. We used the same animal housing and monitored BPA levels in those cages, bottles, etc. And we used a low phytoestrogen diet, and again monitored the BPA and set less than 5 parts per billion as a requirement. We use the automated gavage system, which is, I think, not in common use everywhere, but the animal is taken out, weighed, the scale is connected to the computer, which calculates the dose, and then the dosing pump automatically administers that dose. And we use multiple pumps to try to keep from cross-contaminating. Okay. So there was an issue, an unexpected issue that had developed with background exposure. As I said before, we knew that there was a background exposure from the feed, and based on the levels that we measured in the diets that we used in this study, they varied from 0.03 to 0.2 over the course of the study. We also, at the end, when a tool became available, the standard for BPA glucuronide, samples from the 90-day study were looked at. It was known that using a hydrolysis method to measure BPA, it was difficult in the lower dose levels to distinguish between BPA that was released by the hydrolysis and BPA that might have come from external sources at the low dose level. And when BPA-G became available, it was possible for Dan's lab to indicate that in animals that are 90-day chronic, we did have a background level of bisphenol A glucuronide, which means there was exposure to the animals approximately equivalent to that at the lowest dose levels. We were unable to do this. We've learned this after the study had started. We were unable to do it with the animals in the Clarity study, except for later animals, which I had mentioned before that one investigator had asked for use of the 250,000 dose. So that was very early on in the study. We knew which animals they were. We're not able to measure those, but we did in the animals that were already housed with the lower dose, 25,000, that was the high dose in the chronic study, found that there was no cross-contamination. In the core study, we dealt with this by doing a sensitivity analysis, which has eliminated the animals that were co-housed with the high-dose BPA animals, and we found no discernible impact. Okay. I wanted to say a few words about the study, the conduct, the way we conducted the analysis of the data. The core study, histopathology, was conducted by a study pathologist. He did not do the reading blinded. He compared against the control at all stages. This is a standard NTP practice. All of this is a standard NTP practice. His findings were then placed in a draft report, which were evaluated by an independent quality assessment group, which consisted of 4 veterinary pathologists who reviewed the findings And they, usually, this is one aspect that's unusual. Usually there's a subset of each tissue that's examined. In this case, there was 100% review of the slides from the reproductive tissues in males and females. These results were then, any potentially treatment-related lesions or any disagreements in diagnosis between the study pathologist and QA pathologist were then submitted to another group, a pathology working group of outside pathologists not involved in the study, and these ratings were done blinded. And the results from that pathology working group were what went into the final pathology report. A point to be made about the way we did the statistical analysis. We analyzed the data within sex, dosing arm, and sacrifice time. Litter was the unit analysis in all cases. We did not use litter mates. We compared, we did BPA and EE2 dose groups compared to controls separately. The non-histopathology tests were two-sided and corrected for multiple comparisons for the most part. Histopathology analysis was done consistent with the way that the NTP does these analyses. They were one-sided, not corrected for multiple comparisons. So there were very non-stringent conditions. 0.05 was set as a significance level. Sometimes there's a different significance level of 0.01 for common tumors, and 0.05 is kept for rare tumors. In this case, we used 0.05 for all. So the final pathology report did not suggest that there was any treatment-related lesions. They said they were variable. These lesions were common lesions of aging in this rat, and they're variable across dose groups. So in the final report that we reported, lesions driven by the statistical tests alone. And I'm coming to the end because I'm not going to present the data. It has been, we released all of the data and a draft research report in February of 2018. And I think the releasing of all the data at that time probably wasn't of any use to the general public, but there are a lot of groups interested in those data, and were able to look at them on their own, as well as our interpretations presented in the draft report. The NTP established a peer review panel, went through conflict of interest review and so forth, and a peer review meeting was held in NIHS in April 2018. We took the comments received from the peer review panel, and also took consideration of public comments that came in, and revised the report to produce a final research report, and that has been completed, and it's going to be posted on the NTP website at the end of this month. So that's all I have to say. Thank you.
Thank you very much for a concise description of a very complex matter. A nice example how academic research and regulatory research can work together. We will hear more about this in the subsequent presentation, but we have time for one or two questions. There's one right there, please. The microphone is coming.
Hi, thank you for that very good summary. I also just wanted to make a point that the grantee data is also available, as a lot of it is published and some of it was summarized in a review article last week. If people are interested in taking a look at that, and all of those data are also, I believe, going to go live at the same time as the core study. Maybe Nigel can clarify that, but the raw data, I believe, is coming out in a week. But it's been reviewed in basic and clinical pharmacology and toxicology, and there's a lot of overlap in some of the dose groups, and so I was curious to know your thoughts on those overlapping endpoints in some of those dose groups.
The overlapping endpoints in the dose groups you're talking about between the grantee studies and our study. Yeah. Well, our interpretation was, and I think, and I gave a talk last week, or at the peer review actually, made a heat map of the effects, and it just seemed to be that, actually I haven't had a chance to look at that review article yet, so I don't know which endpoints you're talking about, but our judgment on our study was there was no we used, looking within the organ or within the organ systems to see whether there were responses, consistent responses that would explain the changes, statistically significant changes that we saw, and we didn't feel that that was the case. So, I mean, certainly we have to reconsider all the data.
Write one more there, and then we go on.
Sorry, now it works. Thank you very much. Ninja Reinecke from ChemTrust. I understand that the Clarity initiative was also an idea to look at the guideline studies and the grantee data together. So in particular for the upcoming evaluation within EFSA, it would be really important to have some of the lessons learned from both, from the academic studies and the guideline studies, and what it actually means. So will we see some kind of integrated analysis of the data?
I think Nigel may be addressing that in the next talk, but we did, these reports were done in parallel, so we produced our report, and many grantees have published, and some I think are going to be publishing. So, I mean, all of those data should be available, whether they'll be write-ups or papers to go with all of it, I don't know. But I think Nigel may be able to speak more to how that's going.
Thank you very much. That's a beautiful segue to go over to the next speaker. Thank you very much. So now we have Nigel Walker from the National Toxicology Program at the National Institute of Environmental Health Sciences. And he will speak to us on exactly the point, integrating regulatory and academic investigations in hazard assessment by the USNTP, lessons learned from the Clarity BPA project. Nigel, please.
Thanks, Kelly. And I'd like to thank the organizers for giving me the opportunity to kind of segue on to Barry's talk. It's a complex study, and indeed, it takes 2 talks to kind of describe a complex study like this. So you'll see some overlap, but hopefully not too much, and I'll skip over those where there's too much overlap. So I'm going to talk more about just the whole approach of integrating regulatory and academic investigations. I'll talk somewhat about Clarity, but also about some of the other kind of consortia we've done at NTP and NIHS, really trying to bring these 2 communities together. First of all, I want to declare that I have no conflict of interest, and these are my views. I work for the federal government, but these are just my thoughts about things at this point in time. So just to give you an orientation for the next, I think Gelly gave me 20 minutes, maybe 25 minutes. I'm just going to give you an introduction to some of the background on how we're approaching some of these consortia. I'm going to give you an overview on several other NIHS programs where we've tried to integrate academic and regulatory science on dioxins and nanomaterials. Then we'll get into looking at some of the lessons learned from Clarity and how that worked, and then some overarching lessons learned. So, this audience is probably very familiar with the whole context of risk and hazard and exposure, and what we're really talking about in terms of these integrated assessments are really about the hazard identification, characterization, and the dose response. We're really not, in the studies that we're doing a lot of exposure assessment work, so I'm really going to focus more on the hazard characterization aspects of these programs. And these are all layered on a classic paradigm that we've used in environmental health sciences for the last 20 years. This slide is actually looks a bit like one of those AOP slides, but this goes back to actually about 1995 from both mine and Angelica's former boss used this kind of slide. And it's really that you go from source all the way to an apical adverse effect. And when we think of hazard characterization, we historically have really focused more around the more apical responses and less so much on some of those early biological effects. And this was the topic of several of the talks yesterday about can we do something better by looking earlier in the paradigm of development of those effects. And that's really, we need to move to that direction because we already are using different levels of biological organization in identification of hazard and use of those hazards for regulatory decisions, all the way from cholinesterase inhibition, all the way up to neoplasia. And so the idea of a lot of these investigations are, can we layering different layers of information so that we can make a more robust evaluation of the hazard, not only focusing on the either non-neoplastic responses or on the neoplastic responses. And it's useful at this point to understand exactly how does NIHS, which is part of the National Institutes of Health, which is the primary funder of biomedical research in the United States, how do we actually do funding of research? Because it's somewhat different than how the EU does it, and how the NTP interfaces is also slightly different. So as Barry pointed out, the Clarity program was funded by the Division of Extramural Research and Training. This is the organization within the NIHS that gives out grants to individual researchers. There's also work that goes out under the Superfund Research program, and also worker training programs. In contrast, I'm part of the National Toxicology Program, which is one of the intramural divisions. And there's actually two different types of intramural. That is the intramural research, which is more investigator-initiated. And the National Toxicology Program is more famous for its more contract-based research and testing, conducting studies under GLP, which is more similar to the kind of research that a pharmaceutical company or a chemical company may do for registration of commodity chemicals. And so when we look at trying to integrate programs, what we're doing is actually bringing together immediately two different types of divisions that have different ways of looking at how they do research. And to address, I think the last person's question was about integration of research. This is actually the most critical step in any of these situations because, first of all, different types of research programs produce different types of adverse effects, all the way from molecular, all the way to apical responses. There are a variety of different test systems, different experimental models, and there's also lots of different layers of data trust, as I like to call it, be it the use of guidelines in GLP versus more investigator-initiated research in academic laboratories. And so bringing these two together at the very end is actually the most critical point in this stage. And so here I'm using the concept of systematic review as a way in which some of our programs are being used, but it's really that to convey that sense of the integration is important. And so what we'd like to talk about here is bridging the gap, because one thing we should remember is most of the work that is funded by NIHS and the NIH is really this investigator-initiated research. 90% of money that is dedicated to NIH goes out to the academic researchers throughout the US to do essentially this generated from the roots up, the bottom-up kind of research. And the key thing there to remember is that innovation is critical and the flexibility is critical. The ability to follow one's intuition and to follow new areas of research very quickly. In contrast, what we call more regulatory science, which is what we do at the National Toxicology Program, consistency and acceptance is one of our driving values. We want to make sure that our information is used by regulatory agencies both in the US and around the world. EFSA is one of our primary stakeholders in Europe for our information. And so these two different viewpoints on research create two very different communities that have very different value systems in what their drivers are to generate data and have different pressures placed upon them from those different viewpoints. And so how can we design programs that really bring the utility of both the academic and the regulatory science research together into this regulatory context? So I'd like to pivot now and move on to some of the programs that we've done over the years, because Clarity really only represented one of the more recent programs we've tried and that I've been involved with. And so the first one I'm going to talk about is the dioxin TEF program. Not that we want to talk about dioxins, because I gave up working on that a long time ago. But back in the day, and this was in the late 90s and the early 2000s, there was a key regulatory question about dose additivity of dioxin-like chemicals for risk assessment, primarily for cancer outcomes, because that was the driving regulatory issue at the time. And so what we had back then was a series of studies that I was leading, where we actually had 7 to 8, I think it was 8 different, in the end, chronic cancer bioassays, really testing the hypothesis of dose additivity for cancer. And so as part of that program, we put together, with the Extramural Research Division, a series of what we call RO3 grants. These were very small studies, proposed to use animals off those main studies. And it was really to look at, in a similar context, endpoints that we wouldn't normally have looked at within the context of doing a classic 2-year GLP study. In this case, the outcomes from those were publications on oxidative stress and DNA damage, hepatic transcriptomics, and intestinal hormone gene expression. Now, the strengths of that particular program was, again, we had controlled exposures. There was a whole series of interim time points, so you could look at pathogenesis. There was a well-known mechanism that we were looking at. So the selection of grantees that came out of that was very targeted to people that were working in the dioxin field. But a key challenge with that was that there was really no coordination of the proposals. The RO3s were somewhat random. They weren't targeted to a given question or specific area. It was really driven by the investigator's interest. And again, there was no involvement of the grantees at that point in the study design. We'd already designed the studies. It was, here's some samples, do you want them? There was no central collection of data. There was no requirement for transparency of submitting that data to any central repository. And the RO3 was a very small grant, so it severely limited the amount of work that could be done on that. So there was a lesson learned there. So then more recently, starting in the late 2000s, we had a whole series of nanotoxicology consortia. Prior to this, there was a smaller consortia, but then I was involved with this bigger consortia called the Centers for Nanotechnology, Health and Implications Research. And this was really driven by a lot of activity at the federal level in the US, addressing research needs that had come out in terms of looking at the environmental health implications of nanomaterials. Given the diverse and physical chemical properties, there was a real need to engage the innovation that would come out of academic research into this, to aid in developing risk models for applying those in regulatory acceptance of nanomaterials that were coming forward. The difference this time was that it was a different kind of mechanism for funding. This was a U mechanism. What that does is it allows much closer involvement of the federal staff involved in funding the research with the actual grantees. Normally, there's a lot of separation and not a lot of control. You basically let the grantee works through their projects with very little involvement of federal staff. The U mechanism allows a lot more closer involvement and the creation of a consortia. And so in this case, there were 5 different large centers and 3 smaller centers. Each center had both an in vitro, an in vivo, and a mathematical modeling component to integrate all the information. And a key, the shared activity here, rather than it being animals off a core study, was the sharing of resources, particularly the materials, because a key problem at that time was the lack of good physicochemical analysis of different nanomaterials. So we were part of providing some materials. We were involved with, this is the NTP, involved in making sure that the kind of areas that the grantees were studying were the same areas that we were doing the longer term guideline studies on. Some of the focus areas were on dosimetry and primarily pulmonary toxicity and looking at genetic impacts. So the strengths of this particular approach was the understanding that this U mechanism was a much better way of actually working with a grantee consortium. There was closer involvement, there was regular meetings, the creation of a central collective body of researchers together trying to feeding off themselves of learning how they were working, of developing new methods and stuff. Again, the shared materials across the consortium was a strength. Again, the limitations here was, whilst there was a well-established federal research plan on nanomaterials, the regulatory staff weren't involved in actually selection of any particular research project. And again, there was only a loose connection, therefore, between the actual studies that were done and the specific projects of a given research consortium group. And in this situation, there was no centralized data collection or an a priori expectation that data would come out at a specific time, or a data sharing plan. So that was a very key lesson learned, because there was a lot of scrambling at the end, trying to pull data together. So now I'd like to move on to a more focused effort that we've been talking about, that Barry had introduced this morning, about Clarity BPA. I probably can jump through these because there was a lot of commonality about the background of BPA. I don't really need to tell you all about this. But from an NIHS point of view, the bisphenols as a class is really as an exemplar for many, many issues that we've been struggling with at the NIHS and NTP for many years. And that is, what is actually endocrine disruption? You ask, I'm sure if I ask this audience, what is an endocrine disruptor, I would probably get a good 10 different answers from different people, from different perspectives. And the key question is, how do you actually address hazards posed by these compounds from an a priori point of view? We do a lot of looking backwards, retrospectively at known compounds, and really explore that. But a priori, with a brand new compound that might come to NTP, or you've identified in like Tox21 some endocrine activity, how do we then actually explore the true hazard of that? What is low dose? This has been an issue for the NTP since I've been there 20 years, and we've been discussing what is a low dose effect for the whole time. The issue of shape of dose response. Is there non-monotonicity, or do you have monotonicity of responses? How to integrate those kind of data, which is the topic for today. And then how do you actually do hazard assessment for structure-related compounds efficiently? Because what we see very frequently now is the identification of hazards of one member of a class, a repivoting of use to different members of the class that have data poor information, and trying to read across to those new information. But how do you actually do that in a rapid fashion? So the ability to add new information to guideline studies that could help identify a more faster way of doing things is really needed. So Barry already mentioned this, the creation of the Clarity BPA project, developed between NIHS and FDA, and the goal is really to, again, address some of these issues about the knowledge gaps and scientific uncertainties, and to interrogate endpoints not traditionally measured. And it's also the reason why I'm particularly engaged in this particular project is, again, this idea of a collaborative approach that we can make better use of the federal resources that we use, particularly for investigating complex and controversial chemical exposures. Barry already mentioned this, that there was the development and the evaluation of feasibility and the involvement of the grantees in the core, the final core study design. And the fact this is a 2-year study, the three key, so this goes back to the question I think one lady was asking, what are the key components of this whole program is actually threefold. The core study, and that was what Barry mentioned this morning. This is the backbone GLP study from which animals were then sent to the grantees. But that was conducted like a traditional GLP 2-year study with interim time points. Then you have the 14 grantees following peer-reviewed submission of specific endpoints that would not normally be addressed, looking at much more in greater detail, but using sibling animals born to the same dams that were in that study. But the most important aspect of this is where we're just starting now, and that is the integration report. This is where the information from the core study and the information from the grantees are now going to be started to be integrated. And this is what the phase that we're actually just starting right now. So people are often thinking we're at the end of this. We're actually at the end of one phase of the Clarity BPA. And so this is just in schematic view, just to show that essentially there's a real close relationship between the animals that the grantees got with the core study. And this is the selection of all the grantees that are involved. The key point here is it's a wide variety of grantees looking at a wide variety of different endpoints. Several of these in particular were highlighted in the NTP report for the Center of Evaluation for Risk to Human Reproduction that identified key knowledge gaps, particularly surrounding neurobehavior, prostate cancer, mammary cancer. So we were not only addressing key knowledge gaps that we already had, but we're also exploring other areas. I was involved primarily in the program management of this, so there was a lot of oversight in terms of a management team that had representatives from the grantee groups together with FDA, NCTR, NIHS, a whole series of guiding documents and data agreements. A key aspect was a federated database here. This was right from the get-go. We knew that the information coming from the core study and from all the grantee studies was going to go into a single database that the NTP was managing. And we actually were providing core federal resources to manage that. The whole goal there was such that at the end of the study, we would be able to easily make all the data available rather than each grantee group having to try and do that themselves. In terms of data availability, we have a website on the NTP website. This is where you can come to get all the core study report and the data. There are links there to each grantee, their publications, and what we call the primary raw data sets. This is going to be going live on September the 28th. And again, this is coming to the end of what I think is a very key phase, which is the data collection phase, and that then begins the data integration phase. We want to make sure that the data's out there, so it's not only us doing integration, but anyone can actually start to do that integration. And we're already starting to see that with some of the grantee groups starting to pull together data from these different studies. So just finally, the lessons learned from both Clarity and some of these other projects. A key thing here was the scheme for how we do peer review at the NIH. The fact that there's no involvement of the regulatory end user in either the problem formulation or the direct involvement with the grantees to tailor those particular proposals. And the problem there is that you get proposals that aren't specifically aligned to a regulatory question. And so a key limitation there is that was exactly what happened here. We didn't have the involvement of the regulators very, very early on. Another limitation was academic investigators were forced to use a model that wasn't something that they'd used before. So the issue of reproducibility suddenly raises its ugly head, because now it's not the same model using a different system, and so it may not necessarily be optimal versus past experiences that an investigator used. And another limitation was this centralization of the sample acquisition. I think Barry pointed this out, that it took a lot of resources to do certain acquisition of samples, that in some cases took a lot of coordination, in some cases we couldn't do it, and so again, there was limitations there. But the real strengths of the Clarity approach that I think I've taken away from this is, hey, having that identical exposure conditions really removes some of the concerns that have been articulated about the actual exposure conditions for some of the grantee studies that had not been used by FDA earlier on, or NTP for that matter, in some of those early assessments. A key aspect was the blinding of treatment for the samples that were sent to the core, to the grantees. That way there's no, again, this issue of risk of bias. When you have blinded samples, blinded to treatment, the data is what it is. And the way it worked out was that the data was sent into NTP. There was a team there where we to ensure that all samples were being collected. Somewhat like GLP, there were explanations of why a certain endpoint wasn't collected or an animal was missing. And then once those had been resolved, then we decoded and sent the data back to the grantee. And again, this requirement that all data be collected in a centralized repository really, I think, has facilitated this, because we're now at the point where we can just easily kind of make that release. And again, a key aspect of this was an a priori decision and an agreement by all the grantees right from the get-go that all the primary data from these studies was going to be made available. This isn't something that we kind of decided at the end. It was built into the original design and intent. So looking forward, how can we, in new programs, maximize the utility of such of these? Well, I have to say, I think I would probably focus more on less resource-intensive consortia. This was the biggest program that NTP has ever done, along with the extramural. We had 14 different grantees. The size of the study was equivalent to like 7 NTP bioassays conducted simultaneously as opposed to being staggered. So it really created a lot of pressures on the actual conduct. But the most key aspect, I would say, would be this issue of the problem formulation being targeted early, direct involvement and communication between the end regulatory user and the academic scientists. This will really lead to a closer alignment between regulatory data gaps and the studies that are proposed to meet those. And when you lay those out there more closely, I think there's a much clearer linkage. And as part of that, I think one of the aspects is we need a more flexible funding schema. And there are some of those in the US whereby you can actually go create a group of researchers, and then you actually say, okay, forget what you proposed. You met the bar to get into the consortia. Now let's design exactly what we should be doing in collaboration with regulatory partners, and so you can more flexibly adjust direction and the goals of a program, rather than being held to an original proposal that was provided in the absence of that direct interaction. And I think that's the end, and I would like to thank the organizers for inviting me, and thank you for your attention. And if you have any questions, my email's there, and I'll take any questions here as well. Thank you.
Thank you very much, Nigel. Also very nice to see the lessons learned, the evolvement over time, and I think the key word here is consortia. So I would like to remind you that the app is open. You can also submit questions online, but do we have any questions from the room? Please. Yeah.
It's so bright. Oh, Maurice.
Hi, Nigel. Incredibly impressive from a research management perspective. It's something here in Europe we could really think about more in terms of that integration you talked about. But I'm just wondering, this is, in my understanding, 100% in vivo study.
Yes.
Considering that the last couple of days, we've been talking about new approaches and particularly using non-animal approaches for scientific reasons. What's your? I had to ask. How do you feel about that?
No, actually, what you're talking about, can we develop these consortia for more AOP-driven type projects? So we haven't been doing that from the extramural side. The way that extramural has been involved with NTP has been through the development of new approaches through the small business innovation grants. So that actually is the development of new technologies and new tools and new approaches that could be commercialized. Within NTP, we've developed quite a few consortia where we're more collaborations where bringing in centralized resources that can then be provided through the use of chemical sets from the Tox21 libraries. That's the way that we've been using bringing those consortia together, more sharing of materials. It's the one thing that I've read, and again, the deposition of information, and I'll use the developmental neurotox as an example where we had a series of 90 different potentially neurotoxic and developmentally neurotoxic compounds, including some flame retardants and other compounds. Those were shared with a whole series of researchers. The data was then centrally brought back to NTP, and we had a workshop last year, and that's going to be all that information is going to be on the NTP website with data analysis tools so people could go and explore. So the concept of using shared resources that can then be used, and the centralized database, that model we're extending to other AOP-driven kind of projects as well.
Thank you very much. Nigel, we have a question here. Microphone, please, here. If you just turn it on up here. What? we get another one.
Thank you. That was a very nice presentation, and you know, it works in somewhere like the US, where you are kind of legislated to work together. Do you see a greater difficulty for us here in Europe, where we have different countries working together? I mean, how do you see such a thing developing here?
Yeah,
do you see that more complicated?
Yeah, I know that there's been a lot of calls for, you know, creation of an NTP-esque kind of organization within Europe, where you've got central, you know, what we do at NIHS and NTP is the idea that there's a central place that's resourced. And, you know, when you come to data, databases for data and informatic tools to use on those data, if those are distributed too much, then once that funding dries out, you then get databases and metadatabases bringing together databases. And so if there's one thing I would encourage Europe to do, it would be to create a resources that can at least be providing those central data platforms where people can come so they maintain longevity. Because we saw that with some of the nano consortia. Initially, the very early databases were distributed, and there was very limited resources to maintain the longevity, and they basically disappeared. And then people are having to pull data. That's a waste of resources. And so to that extent, I would encourage that.
Thank you very much. So more important messages, data sharing, transparency and funding. I mean, the Clarity BPA, as you said, was just a tremendous effort. If the willingness is there to work together and the funding is there, a lot of things are possible. Are there any further questions from the audience? Yes, please. Would you please introduce yourself? Thank you.
Ninja Reinecke, ChemTrust. During the peer review process of the draft NTTP report, the Endocrine Society had raised quite a lot of concerns regarding the use of historical controls. There were questions around the non-monotonicity considerations and some of the statistics. So how did you address those, and how will this continue to play a role in the integration of the data?
That's a good question. Yeah, so the way that normally the NTP handles peer review comments, just in general, is all those comments are taken by the authors of the report and integrated in, you know, used to actually make any changes to the report. Now, the thing to remember here is that, as both Barry and I pointed out, the goal was to look at if you did this by a classic GLP study design versus not having the grantee, what would you have concluded? Because that's where the whole value is. When you add that grantee information, would that have changed the conclusion? And so when we were looking at how to make conclusions from the core study, it was very much a classic NTP study interpretation. And so, you know, some of the issues related to historical controls were, you know, challenges, but they're part of how we'd normally do the business of NTP. The NCTR has a different kind of historical control system, and so we've tried to put a little more clarity, pardon the pun, in the report about that. But the whole point was not to try and integrate other findings at that point in time. But that will come forward in the integration phase of how to actually, what are some of the challenges of bringing those two data sets together? You know, the use of historical controls only for neoplasia and not for other endpoints. That's not something we normally would address in the normal NTP report.
Thank you very much.
Okay.
Thank you very much, Martin, once again. So now we shift gears a bit. We heard a lot about animal experimentation, academic, and linking academic and regulatory research. And now it's my pleasure to introduce again Thor Haldorsen from the University of Iceland, and where he's a professor at the Faculty of Food Science and Nutrition, and he works on nutritional and environmental epidemiology. And he will present on the use of epidemiological studies for setting health-based guidance values. Thor, please.
Yeah, so common with I'm not going to talk about BPA, and common with the two previous speakers, I'm not going to present a lot of result, and therefore my talk might be a little bit boring, but what I'm going to talk about is setting health-based use of epidemiological studies for setting health-based guidance values. And I'm not a toxicologist. The risk assessment procedure is relatively new for me, but as I identify it here is that we are talking about identifying the critical outcomes in the critical study, and the most part of my talk will be about determining the point of departure and how you might do that in epidemiological studies using benchmark dose analysis. And the question that I'm going to ask again, can the same methodology be used for human observational studies? And to answer that question right away, I think the answer is yes. It's less complicated than some of the pesticide studies that I was showing you yesterday anyway. Now, but it's okay to ask yourself and then sort of start as a baseline. So, I mean, controlled animals experiments, why do we use something else? And this is the standard for risk assessment, and this is where the benchmark dose has mostly been used and developed around. Because the controlled setting is such a major strength that you cannot match it in human observational studies when studying adverse effects. And of course, using the animal models is more time and cost effective than the large-scale epidemiological studies. I've been, for the past 10 years, working a lot in life course epidemiology, and that means that you're working with people born in the 1970s, 60s, and up down to the 1930s. And you find some environmental factor that is correlated with some health outcome, but by the time you realize that this is the case, the world has changed and the same environmental factors are no longer in place. And the same with chemicals. I mean, if you're studying chemicals in epidemiology, they have been in the environment for many, many years, usually. And of course, the animal studies are subject to far fewer ethical constraints, but the problem is, again, that you're studying animals, but not humans. Now, looking at the sort of classical risk assessment, the luxury of working with controlled studies, in my view, often results in large weights being given on a single study or a single outcome. You talk about most critical study, the most critical outcome. And then you do a benchmark dose analysis on that and derive the health-based guidance value. But working with humans, my sort of impression is that the study population, if you're looking at children, adults, the elderly, and the environment, partly determines the dose response that you see. And just taking an extreme example, if I take adults who all have the age of 25 and they are all males, half of them are normal weight and they are very fit, the other ones are overweight or obese and pre-diabetic. And if I put them into an oral glucose tolerance test, the dose response curve that I get is absolutely different. And that is the environment. So when I determine the point of departure, I mean, how is that done? And I'm just going to sort of, now what I'm going to do is just to compare how it's done for animal studies and how it maybe should be done for human studies. Now, we are all agreed that the benchmark dose is the sensible approach, and I think it's a fascinating, it's the correct way to drive a point of departure, because instead of looking at individual data points, you basically use the whole information that you have from the study to fit a curve through the data points or the dose response curve. And it takes biological relevance into consideration because you decide a priori what is my benchmark response, what is the point of departure that I consider relevant. And the model fit provides you some indirect information on the study quality, on the uncertainty, because if there's a poor fit, that's a clear indication that something wrong with the study. And also the model fit gives you some indication on biological plausibility. So you're combining all these things, and from a statistical point of view and biological point of view, using the whole information is always better than looking at individual data points. And there is a very nice EFSA guidance, and I was sitting in the scientific committee last term and I read it and I enjoyed it. I usually don't enjoy guidance documents, but there was something that I finally understood, and it was nice. But it's, sort of when I read it, I immediately, my perspective was, yeah, this is for the animal studies. I mean, there are common things that we can extrapolate to human. There's the use of maximum likelihood advocated. I have no problems with that. They use model averaging for quantile data. That might be sensible. It takes away individual modelers' decisions, but maybe in a regulatory perspective, then you don't have to take any decision, and then there is maybe nothing to quarrel about, but there are limitations to this, but this is sensible in this respect. There is a restrictive set of models that is advocated, and that might be fine for animal studies, because in the guidance, and there's a separate publication, you modeled about 500-600 different studies and really showed that this works. But I must say that some of the models, like the continuous models, were models that I hadn't seen since I was studying statistics in school. These are not models that we conventionally used for human epidemiological studies by convention, but they can perhaps be used, but we have not been modeling 600 human studies to test that. But I think it will work anyway. Now, and that is my perception, that the existing guidances, and we start with the EFSA guidance, they are designed for controlled animal experiments. And in a 40-page document, there is about half a page, or 1 page on guidance and half a page with example. And in this half a page, they say the analysis of human dose response data can be more complicated than that of typical data from animal studies. And the reason, due to confounders and imprecision in exposure estimates. I mean, we deal with confounders all the day. It's not very complex. Imprecision in exposure estimate, it varies, but it more or less creates a little bit of misclassification. You lose some information, but in principle, that's not a problem. And then they say, which is right, that in principle, the BMD approach would also be applicable to human data. Then you read the benchmark dose technical guidance from the US EPA, and slightly different view. They say opportunities for modeling human data are more limited, and I will get back to that in the next slides. They say studies are less standardized. That's absolutely true. And the modeling is often involves additional considerations, such as adjusting for covariance. So that is not problematic. And then they say sometimes human toxicological data are reported in ways that are similar to animal data, and not conducted but reported. In these cases, the guidance document would be applicable. But let's stop around this opportunities for modeling human data, because that was touched upon, but no further guidance is then given in these documents. Now, there was some presentation here on the first day when they were talking about transparency as a mask, and the US EPA not proposing on sort of not looking at human studies if individual participant data couldn't be delivered. But let's start with the first point. Previously, one of the complaints has been that the way data is reported, but previously the benchmark dose analysis has not been used in human studies. There are more and more publications coming out. It's interesting, it's being used. But if there's no reason to use anything, then there is no reason to report it. But I think if we make the message absolutely clear and can reach a consensus how things are done, it's no problem to report the data in a way that data can be modeled. On the individual participant data, from a statistical point of view, I can't see a logical argument why you would need individual participant data. If you need individual participant data, then you also have to do your homework, because animals are not humans, and animal welfare does not cover the health of the animals and their private information yet. However, when I'm working with data, if I want to ask children in Iceland if they eat apples, I have to go to the ethical commission and I have to apply for it. Now, if I want to share the data, I have to get a permission for that. Working with data in Iceland, it doesn't take a long time for me to identify a cancer case in the northwestern part of the country who's a female with a certain type of cancer. Working in Copenhagen, it's also possible. Now, there are good reasons for these data protection rules. So if the US EPA, EFSA, or anyone wants to have individual participant data, then you need to get your act together, have a secure server, you have to have legal contracts, you have to have contracts how the data is delivered, who is going to use it, when it's going to be deleted, are you going to do any additional analysis? These things have to be in place, and then we can talk about individual participant data, just to make that clear. But that was the boring stuff. So now let's get into what characterizes the use of BMD modeling for animal studies, seen from an epidemiological point of view. Now, you have the control dose, which is very nice. There is absolutely, and you also dose the animals by feeding them, not by measuring internal concentrations, which are sometimes problematic. You have large exposure range seen from an epidemiological point of view. Now, imagine this was sugar or added sugar or free sugar or whatever it is. Then this study would be giving something between 0 grams per day and 1 kilo per day over a couple of years, and that would be absolutely unethical. But if I would do that with sugar, I would get a very nice dose response curve when it came to fasting blood sugars a couple of years later. And this is not the case in human studies. We are not studying that range. You also have sometimes large spacing between doses, and from my perspective, I think this is one of the reasons why the benchmark dose modeling is so nice for these controlled experiments, because instead of looking at no-well and low-well, you fit the model through the data and decide the point of departure, and you don't have to stick to this really large interval taking the low-well. So that's one of the strengths. And of course, variability is small. You're studying genetically inbred brothers and sisters, and you have very controlled conditions. And then you have the safety factors. And I really don't understand sometimes, this is some report that I found on pesticides. A safety factor of 100 was used for 187 studies, and then for the 26 remaining studies, there was one human study. But otherwise, you deviate and you can have a factor of 200, 300, 500,000, 2,000, and 10,000. And if you have, I assume if you have so large uncertainty factors, then that you didn't have much information. But if you apply large uncertainty factors, it doesn't really matter what you're quarreling about here. I mean, everything is just lumped together. So I really don't understand sometimes why we are quarreling about modeling. But sometimes it matters. Safety factor of 100, I can go with that. For human studies, you have no zero dose. Now, this is an old example. These are 10 publications by Matthew Longnecker. There's 10 different studies on PCBs, but he plots the LEAP standardized PCB 153, and you have here the sort of the boxes, the sort of the 25th to 75th percentile, and this here would be the lowest 25% of the exposure range. And what you can see is here, if you take a study from the United States, that the lower part of the exposure is around 10 to 20 nanograms, but if you go to the Faroe Islands over here, it's 100 to 300. What you also see is that in some studies the exposure range is very narrow. For example, in this study from the Netherlands, where everybody is drinking the same milk and sandwich and everything is quite sort of narrow. Now, that more or less determines a lot of things when you look at these in human perspective. If you have a theoretical dose-response curve with something related to PCB 153, if you have very low exposure and very narrow exposure range, you will get a zero. You will just be placed here on the dose-response curve. Theoretically, if you are slightly higher, if your starting point is slightly higher, you may up with something that is non-linear. And if you're slightly higher, if you're in the Faroe Islands, you might end up here. So the dose response that you get when you have this depends on the starting point and the range. And what do people then do? They do a meta-analysis where they lump everything together and they take no notice of the baseline concentration. And that is sometimes also my feeling. We are counting pluses and minuses, and if we see differences, it's inconsistent findings. But it doesn't always work that way, but it's sort of on average. Now, in human studies, spacing between doses is not a problem. You're measuring exposure on a continuous scale. And I have a different exposure profile from my wife because we eat slightly differently and we exercise differently, and there's a lot of things even if you live in the same environment, so you get continuous data points. So extrapolating between a no-well and a low-well is technically not an issue, even if you work with quantal data. But the benefit here, the clear benefit is again the biological relevance, determining the benchmark dose, and in EPI-study we tend to be horrible at actually looking at biological relevance. There has been over-focus on statistical significance. So thank you for introducing that into our world. And then again, you have very large variability in our studies. And this is an example of IQ in children and lead exposure. And thank God it is this way, because you can be highly exposed to lead, but if your parents really love you and treat you respectfully, you might have a higher IQ than if you're working in a less unfavorable environment. But there is a momentum that if you have high lead exposure, your IQ will go down. But this is the variability that we're working in. And with that variability, I mean, you can decrease it statistically with a large sample size, but I seriously doubt or question the use of the lower confidence interval of the BMDL being used in human studies to derive a health-based guidance value. Because in the environmental epidemiological studies can range from 50 to 50,000. Animal studies are more standardized, and there's a momentum in a way to minimize everything by researchers who believe in their own findings. I don't believe in all my findings, so I'm more neutral. But we really have to be careful about this, because this is going to be misused. And that is the key issue that I think, because this is less standardized. And of course, you cannot have controlled conditions. We can't do experiments on humans when it is adverse. And we have no safety factors. And then I just wanted to sort of finish this boring presentation with a short example, just to see how we do it in practical terms. I'd like to take the example of serum perfluorooctanoic acid and cholesterol, because there's a very nice study from the C8 population. There was a DuPont in Little Hocking, US, sort of managed to contaminate the drinking water around the factory, exposing a lot of people to relatively high levels of PFOA, which was relatively independent from other perfluorinated acids. Now, there was extensive research. They measured They recruited about 60,000 people and measured serum samples, but some of the studies were not of different quality because they had to go back in time. But the panel of C8 scientists concluded that there was a probable link between PFOA and high cholesterol. And this is how the dose-response curve looks like. And I'm just showing this because this is an example of how epidata is reported. It's not reported by giving you some non-linear coefficients or anything. They break the data down into 10 equal groups. We have 46,294 people in the study. You break the data points into 10 equal deciles, so that means that there are 4,629 subjects plus minus in each decile. And then you plot the mean cholesterol level in each group as a function of the median concentration of PFOA in each decile. So this is quantile data. Reporting the individual data, I don't see anything that would have changed anything here. So this is not individual participant data. But what they do is sometimes we would publish this curve as a relative increase in cholesterol, but here they do something different. They say that it has been adjusted for covariates, the model, so you remove the variation of age, body mass index, smoking, and everything. But they say the predicted cholesterol levels are based on an average subject following these characteristics. It's a 60 to 69-year-old male, not taking cholesterol-lowering medication, never smoked, high school diploma, does not participate in regular exercise, does not drink alcohol, and a body mass index of between 20 to 24. And what on earth does that mean? And I would just like to draw your attention here that the lowest decile has a mean cholesterol of 199 milligrams per deciliter, which was the mean in the study population. It basically means that you have a linear regression model where you have removed the variation of the covariates, and what you then do is that you plug in some covariate values here, and that merges with the intercept, and you just end up with a relative dose-response curve anyway. And which covariates you can create an average male or an average female or whatever you like, it's just setting the starting point on the y-axis. It has no implication for the dose-response curve. But why do we do this in epidemiology? By convention, in the old days where statistical software packages were not that powerful, people did simple statistics. By convention, we want to show in a simple terms, in understandable terms, what the risk is, or what the relative risk is, or how the mean estimates look like, because some medical doctors like that. So it's just different convention. It can be modeled any other way, but referring to my lead figure, if we would show data as it is, nobody could read it also. Okay, and let's go forward with the example. I chose a BMR of 4%. Why did I choose a BMR of 4%? That is a really small increase, isn't it? Because it corresponds to around 75% of the increase in cholesterol observed between normal and obese subjects. It's about half of the cholesterol increase on average that you see from age less than 30 to more than 80. So it is a quite strong dose response, I think, because it corresponds to some of the strongest determinants of cholesterol. And there are two very nice softwares available. PROAS developed by RVM and BMDS. I've looked at both. They both have their strengths and weaknesses, but I found for this occasion that the BMDS was more flexible, and don't ask why. For referring to the US APA guideline, they say sometimes human toxical data are reported in range that are similar to animal data. And when I look at these data, I think this is one of the cases that the word sometimes refers to. And trying blindly to model that, you get an absolutely ridiculous value, because both software packages extrapolate down to zero, because in animals you always have the lowest dose. And this is silly. And then you just need to scale the data, you just subtract, and you define the lowest decile as 0, but not 5.5. It doesn't change anything, you're just changing the, where you put your x-axis, x-axis. And then I get a BMDL of 48.9 when I've added these 5.5 nanograms in the lowest dose, and a BMDL of 4. And if you look at it by hand, it's the same. This is not rocket science or anything. Now, so there's a large difference there. So remember to scale the data. Now, the mean concentrations in the lowest deciles were 5 nanograms, which was the US average in 2000, and that's relatively high today. And there was another study, because I don't like these single critical studies. So I had another Danish study that I could model. The concentrations there were about 2-3, so I extrapolated down to 2 nanograms by calculating what would be the mean cholesterol level there, and then changing the intercept, and that gives me a benchmark dose of 8.5 and 6.7. Here, the BMDL is fine because the precision is so high. But when I model the Danish study, You can see the confidence intervals are quite large. It's relatively insensitive. I can get, based on a model fit, slightly different values, but I managed to get more or less the same levels, but the BMDL is of course slightly lower. So here I have two studies showing the same benchmark response, reflecting that there is consistency between studies, or the same confounding factor for that matter. But in conclusion, I think there are no major obstacles for using human data to derive health-based guidance values. The BMD analysis can easily be performed, but existing conventions may not be directly applicable, and more work is needed. I'm not talking about months of work, I'm talking about minor consensus in how we report data, how we use the BMDL, and I would like to see similar work conducted as with the model selection. Do we have to restrict ourselves to these few models? Lack of individual participant data is unlikely to be a key issue, and if somebody thinks it is the case, please provide me with the simulations. Due to high variability and varying sample size, I don't like the BMDLs, and modeling should not be done for the sake of modeling, because my impression is that toxicologists sometimes like to model, and that is driving the way. We can't use it, it's better to model this study, but it's the end point that is the key. Thank you.
Thank you very much. Quite an important topic, and please allow me to use this opportunity to inform you that WHO, we are currently updating our guidance on benchmark dose modeling. We work together with EPA scientists, we work together with EFSA scientists on this, and it will include a section on guidance on the use of benchmark dose modeling for epidemiological data. And I hope we can involve you in this exercise as well. We have time for questions. May I start with a quick one? You have said, can you hear me? It's a bit, is it better like this? Can you hear me? Yes. You said that you used the both available software, BMDS and ProAS. Did you see any differences in outcome?
Yes. There are some differences in outcome. For the few, I mean, I'm not a benchmark dose model, but I tested a little bit, and I think it was around 10–15%, and it has to do with that the BMDS does not automatically log transform, but the ProAS does. And I mean, both have their strengths and weaknesses, but you know, you can decide to log transform your own data, and that BMD gives that flexibility. But I mean, sometimes we spend hours discussing about, you know, values of no relevance, and I don't want to go into that discussion.
Thank you very much. Any questions from the audience, please? Please. And please introduce yourself.
Yeah, Christophe Roussel from ANSES. Thank you for your very inspiring presentation. I would like to know if what you have presented would also apply for non-tracehold compounds, because for me, what you have presented could be used for tracehold compounds, but usually, for example, for genotoxic carcinogen, when we start to derive point of departure and then extrapolate to low doses, how would you implement your approach for this kind of compound and to calculate extra risk or excess risk? Or
It's a difficult question because I haven't worked with these types of assays, but I mean, my key issues are I can imagine with some of these assays you have high variability, and then I would be a little bit careful, and I would also be careful in I really don't like extrapolations if they can be avoided. That's my, but I must admit I haven't worked with these types of data. I haven't seen a lot of these data, so I'm sorry, can't answer that.
Thank you. Yep.
Hi, Heather Patisol from North Carolina State. Thank you for a great talk. To try to bridge some of the gaps between the animal data and the human data for the modeling, there are some genetically diverse animal models now, like the collaborative cross mouse, where you can still do a lot of controlled study, you can control the dose and things like that, but you have a greater genetic diversity, which might get at some of the variability that you're seeing with the human data. Do you know of some efforts that are using that kind of data to sort of work on these model sets and evaluate some of these models in conditions where you have more of that genetic diversity?
No, that's the simple answer, but I welcome the fact that people are studying more diversity in these models, because I think it would be a nice supplement. Because sometimes we are working on so different scales sometimes that it makes life difficult. Thank you.
Thank you. There was one more question right there also.
Thank you for the talk. A key issue I see in epidemiological data is exposure assessment, because in many cases exposure is not based on biomarkers or reliable biomarkers. Sometimes you use proxy for exposure, and this may introduce a lot of bias. And if you can comment on this.
Thank you. It's a good comment, and I should have said it. I mean, you don't do a benchmark dose modeling on human data unless you have relatively accurate, and then I'm talking about actual measurements in a way. I mean, I can imagine still if you would have a continuous scale self-reported or in an occupational setting, it may be justified, but I would be very careful still. So this is more like, I mean, these are easy compounds like lead or cadmium or mercury or PCBs, dioxin. Yeah.
Thank you very much.
There was one over there.
Here in front.
There's also a gentleman here at the back who has been waiting.
Hello.
In the back first, and then. Would you please start in the back first, please?
Gianfranco Brambilla from the National Institute of Health, Italy. I kindly ask you if in the association between FOA exposure and cholesterol level, there are some clinical evidences about a higher rate of cardiovascular diseases in the exposed group. Thank you.
Yeah, well, on P4 and cardiovascular risk, I mean, I didn't hear the question properly, but I don't want to go much into the causality and what it says, but just point out that, I mean, this is the largest study in the US. You can't do a proper follow-up, so nobody has been able to look at the cardiovascular risk that this may facilitate. Sorry.
And you made the remark during the presentation about the importance about biological relevance. Please.
BFR, Germany. So that's exactly my question also. So I think if you do this, the mode of action must be clear, or more than less, not less, more evidence than less evidence. I think, is this a precondition to perform this? Because the outcome has enormous impact if you don't consider this.
Yeah, but here we are talking about how to do benchmark dose modeling if it was not a presentation on the causality of FIFO and cholesterol.
Thank you very much. We are now a little bit over time, so we're going into coffee break, and I hope to see you all back. And thanks to all the presenters.
Deusa da lenda na proa levanta uma tocha na mão. Todos os homens do mundo voltaram seus olhos naquela direção. Sente-se o gosto do vento cantando nos vidros o nome doce da cunhã. Vai o mundo inteiro num leve leviatã. E aqui dançam guerras no meio da paz das moradas de amor. Ah, pra onde vai quando for essa imensa